Good afternoon. I'd like to thank the organizers for inviting me to participate in this symposium, and I'm sorry that we cannot be together because of the coronavirus crisis. Today, I'd like to talk to you about some work we're doing in my lab to use systems biological approaches to understand the mechanisms of vaccine efficacy. Vaccines have been described as one of the most cost-effective public health tools in history and have saved countless lives from the specter of infectious diseases. Yet, to immunologists, vaccines represent incredibly powerful probes on the human immune system. Why do I say that? I say that for several reasons. First, vaccines can allow you to perturb the human immune system in a very synchronized way. Infections and autoimmunities and cancers and allergies can also perturb the human immune system, but one can never know with any degree of certainty precisely when the perturbation happened. With vaccines, however, you know exactly when the vaccine went into the body. Secondly, the advancement of high-throughput technologies such as transcriptomics, metabolomics, epigenomics have really allowed one to analyze the behavior of cells, genes, metabolites at an unprecedented degree of precision, so that you can really follow the immune response to vaccination with time at multiple levels of the biological hierarchy. Thirdly, is a practical reason. We all get vaccines. Vaccines are administered widely to very diverse populations, ranging from the very young to the very old, to those of us who are susceptible to autoimmunities or allergies, cancer patients. So the incredible diversity of phenotypes, human phenotypes, that is manifest in such diverse populations can be understood by perturbing the immune system with vaccines. Fourthly, vaccines represent a diverse array of viral and microbial stimuli, ranging from live viruses such as smallpox or yellow fever to inactivated vaccines, RNA vaccines, DNA vaccines, recombinant vaccines given with adjuvants, without adjuvants, carbohydrate vaccines. There's a whole array of different stimuli that are used to perturb the immune system. So, this gives us a wonderful conceptual framework with which to probe human immunity and to understand how the host genetics and the environmental factors, as well as the microbiome, impinge on the immune physiology of human beings. Several years ago, we performed a study in humans to use systems biological approaches to analyze the innate and adaptive responses to vaccination with the live attenuated yellow fever vaccine. The yellow fever vaccine, or YF17D, is one of the most successful vaccines ever developed, having been administered to over 600 million people globally and having an efficacy of greater than 99.9%. Yet, despite its success, very little is understood about the mechanisms by which it stimulates protective immunity. So we performed a study, a phase 1 study, in which we vaccinated people with YF17D and analyzed the innate and the adaptive responses as shown in that diagram. In Rafi Ahmed's lab, they analyzed the magnitude of the tetramer-positive CD8 T cells as well as the neutralizing antibody responses. In our lab, we analyzed the early innate response that occurred within the first few days. And in particular, we looked at the changes in the gene expression profiles induced by vaccination, as well as the changes in the dynamics of innate cell populations by flow cytometry, and analyzed the changes in cytokines and chemokines by Luminex analysis. We were able to define an early signature, a molecular signature of transcriptional changes, if you will, that correlated with the ensuing CD8 T cell response and the antibody response. We were delighted to observe that a similar and independent study done by Rafiq Sekali and colleagues came up with pretty much identical signatures to what we had seen before. Furthermore, we used machine learning techniques to define a focused gene list of predictive rules, if you will, that could be used to predict in an independent way the magnitude of the CD8 T cell and the neutralizing antibody response in a completely independent trial that was done some 1 year after the completion of the first trial. So this study represented the first example, the proof of concept of the utility of applying systems-based approaches to defining predictive signatures of immune responses to vaccines. And in work that I'm not going to discuss today, This study has also revealed many interesting mechanistic insights about the mechanisms by which this vaccine is stimulating such effective immunity. Subsequently, we and a number of other labs have extended the so-called systems vaccinology approach to understanding immune responses to diverse vaccines administered to diverse human populations, including those at the extremes of age. These are some of the publications listed in the bottom left that we have published over the last several years, but really this approach is now quite widely used in the field to understanding and analyzing immune responses to vaccines. And some of the vaccines that we've focused on are shown here. I mentioned the yellow fever vaccine, but we've also been focusing on the seasonal influenza, avian influenza, malaria vaccines, and in the elderly, we're looking at shingles vaccines, and in the very young, we're looking at childhood vaccines. Another innovation that's happened in the field is the integrative modeling of multi-omics datasets. In this study, we analyzed the immune response to shingles vaccine, the Zostavax live attenuated shingles vaccine, and we analyzed the transcriptional profile, the metabolomic profile, plasma cytokines, and high-dimensional mass cytometry analysis. We were then able to integrate these orthogonal data sets to construct a multi-scale, multifactorial response network that revealed critical insights about the mechanisms by which this vaccine was inducing antibody responses. So in this talk, I'd like to show you one example of how systems biology can be useful in providing deep mechanistic insights about the human immune system. And I'd like to focus on what we call nature's endogenous vaccine adjuvants, and I'm talking about the microbiome here. And recent estimates are that if you calculate the number of genes in the humanity's microbiome, in other words, in the microbiome of every single living person on the planet, if you count the number of genes you have, these outnumber the number of stars in our universe. So our journey on this project began about 7 years ago when we analyzed the immune responses to the seasonal influenza vaccine using systems biological approaches. We defined a gene signature induced by vaccination early after vaccination, 3 days or 7 days, that correlated with the subsequent antibody response. And in particular, we observed that the expression of TLR5 or toll-like receptor 5 was highly correlated with the ensuing hemagglutinin titers induced a month later. Of course, you recognize toll-like receptor 5 is a receptor that senses bacterial flagellin. It was a bit of a puzzle as to why a vaccine against influenza should be inducing such a gene, and more importantly, why this gene was so strongly correlated with the ensuing antibody response. Jason O in my lab decided to investigate this puzzle, and what Jason did was to analyze mice in which TLR5, the gene for TLR5 was deleted, so-called TLR5 knockout mice, and he immunized wild-type mice or TLR5 knockout mice with the seasonal flu shot, and he saw that, in fact, there was a very profound impairment in the flu-specific IgG antibody response. Interestingly, Jason also observed a similar reduction in mice administered antibiotics or in germ-free mice, arguing for a role for the microbiome in orchestrating the antibody response to the flu shot. Of course, this was an experiment that was done in mice, and the question was, does this apply to humans? And why should one be concerned about this? Well, for many reasons. Firstly, as immunologists, we are curious about the mechanisms by which the human immune system functions and how it's affected by the microbiome. Secondly, from a practical perspective, of course, vaccines are administered widely to people in different parts of the world, and it's now becoming very clear that the microbiome composition of different populations in different parts of the world vary very widely, as shown here. Now, and yet, we all get the same vaccine. There are no personalized vaccines that are given to different segments of human populations. So the question was, do the differences in the microbiome in any way impinge on the immune response induced by these vaccines? To address this question, we performed a human clinical trial. When I say we, this was really the brainchild of a wonderful infectious disease physician by the name of Nadine Rupail. She is at Emory University, and together with her, we designed the study in which healthy human adults were administered a cocktail of 3 different antibiotics, neomycin, vancomycin, metronidazole for 5 days. On day 0, that is 1 day prior to the last day of antibiotics treatment, they were immunized with the seasonal flu shot. And then we analyzed the response for the following year. So these are stool samples were collected all the way up to 6 months. Blood samples were collected all the way up to a year. And we did an extensive analysis of the change in the microbiome, but also the change in the innate and adaptive responses using various different assays shown here. The results of the study was published about 4 months ago, and I'd like to summarize the key findings from the study. The first question we asked was, what is the impact of this antibiotics administration on the microbiome? So we performed 16S ribosomal RNA sequencing to quantify the total bacterial load per gram of stool. And as you can see in red, these are the subjects who received antibiotics. There's a very profound drop in the total quantity of bacteria per gram of stool. And there's about a 3 to 4 log drop. It's about a 10,000-fold drop in the quantity of bacteria. What's very impressive is the rapidity with which this rebounds. Within a week or so, things are back to baseline level. Next, we analyze bacterial products in the stool. In this case, we're measuring the concentration of bacterial flagellin, or LPS, and this was performed in Andrew Gerwitz's lab. And you can see, similar to the total bacterial load, there's a very profound drop in the concentration of flagellin and endotoxin, but again, it's very transient and short-lived. Next, we analyze the impact of antibiotics on bacterial diversity. What's very obvious is that there's a remarkable constriction of bacterial diversity within a few days of antibiotics treatment, but unlike the rapid recovery in bacterial load that we saw in the previous slide, in fact, there's a much more gradual recovery, so that even at day 90 or day 180, the diversity that was observed at baseline has not yet been reached. So this tells us that antibiotics treatment has a very profound effect on the bacterial load as well as the bacterial diversity. However, whilst there's a very rapid recovery in terms of the bacterial load, there's a much more gradual recovery in terms of the bacterial diversity. So the key question is, what is the impact of this profound dysbiosis on the immune response? When we analyze the microneutralization titers induced by influenza vaccination, we observed there was absolutely no effect, despite such a profound effect, on the gut microbiota. Here we're looking at the microneutralization titers against the 3 different strains that are contained in the flu shot, the A California, A Texas, and B Massachusetts strain. This was measured in the laboratory of Hannah Golding and Surinder Khurana at the FDA, and the results were very, very clear. No impact at all on the immune response. Furthermore, we did a variety of different analyses, some of which are shown here. We fact sorted single plasmablasts and measured the somatic mutation in the immunoglobulin V genes with Patrick Wilson in Chicago. We analyzed the T follicular cell responses using flow cytometry and observed no differences. So this initial study was performed in 2015 in subjects who had relatively high baseline titers against the flu vaccine. Subsequently, in 2016, we performed a study in which we selected subjects who had very low baseline titers. We screened many subjects and we selected those who had very low baseline titers, and we excluded from the 2016 study any subject who would have received the flu shot in the 3 years preceding their enrollment into the study, or who would have had influenza infection in the 3 years prior. So what we were trying to do is to select for people in whom immune memory against influenza was minimal or relatively low compared to the average person. When we analyzed the antibody response in the 2016 trial, that is, in the trial in which we had selected for subjects with very low baseline titers and selected for subjects who had not received the flu shot or had been infected with flu in the 3 years preceding their enrollment, There we saw a profound impact on the antibody response. When Galit Alter's lab measured the H1N1 specific IgG1 binding antibody titers, there was a very profound drop in the antibiotic treated people compared to controls. This was confirmed in Hannah Golding's lab by Surendra Khurana, and also in Hannah's lab, when they measured the microneutralization titers, there was a very substantial effect in the antibiotic-treated people. Furthermore, we also looked at the IgA H1N1 specific binding antibodies, and consistent with what we saw with the IgG1, there was a major impact on the IgA H1N1 specific antibodies also. So clearly, antibiotics treatment had impaired the antibody response against influenza vaccination in subjects with low baseline titers. What about the innate immune responses? We analyzed this using flow cytometry of analysis of innate cell populations in the blood, or using transcriptional profiling. And what was immediately obvious was that transcriptional modules involved in inflammation, so in other words, these modules contain many genes that orchestrate the innate immune response and the inflammatory response, were highly enriched in subjects who had antibiotics treatment. Why this was of great interest to us was that in a previous study that we published about 3 years ago, in which we had analyzed the transcriptional changes to influenza vaccination in healthy young adults versus elderly, we had seen that these very same modules that we were observing in the current study were highly enriched in the elderly subjects in the previous study. In other words, antibiotics treatment of healthy young adults had altered their transcriptional profile to vaccination to make it look like the transcriptional profile of an elderly person. In addition, we analyzed changes in the plasma metabolome using mass spectrometry. And in this principal component analysis, each line represents the metabolic trajectory of an individual, and each circle represents a time point. So, for example, you can see take any blue line and you can follow the line through its day 0, day 1, day 3, day 7. Interestingly, the blue lines and the red lines segregate very clearly. The red lines represent people who took antibiotics, and the blue lines are people who did not take antibiotics. In other words, antibiotics treatment has dramatically altered the metabolic profile in the plasma. In order to understand the metabolic pathways that might be perturbed by antibiotics, we use MummyChog, which is an analytical program developed by Shuzhao Li when he was in my lab as a postdoc. Basically, MummyChog allows you to go from untargeted metabolomics data to a pathway. And what was very clear was that there were major biochemical pathways that were perturbed by antibiotics, but at the top of the list was bile acid biosynthesis. To verify whether bile acids were in fact changed by antibiotics, we performed chromatography analysis, and we could see that in fact there was a major change in secondary bile acids, shown here. And this is one example of the extent to which secondary bile acids are changed. We're looking at the lithocholic acid here, and you can see there's about 1,000-fold drop in the concentration of lithocholic secondary bile acid in the plasma. And this is quite long lasting, and then at 3 weeks or so, things are back to baseline. So major change in secondary bile acids. Now, when we analyze the change in secondary bile acids shown here on the y-axis versus the total bacterial load shown on the x-axis, or concentration of flagellin or LPS, you see that there's a very profound and highly significant correlation. So in other words, the more bacteria you have in the stool, the higher the concentration of secondary bile acids. Now, what is secondary bile? Well, bile acid biosynthesis begins in the liver through metabolization of cholesterol, which gets converted into primary bile, which gets transported through the gallbladder to the intestine, where it gets metabolized into secondary bile by the microbiota. Secondary bile can bind to 3 known receptors, one of which is shown here, FXR or farnesyl X receptor, and mediate a range of biological activities. We analyzed the expression of the genes encoding the secondary bile acid receptors using the ImGen database, and we were very surprised to see that myeloid cells in human blood, including dendritic cells, monocytes, and other myeloid cell subsets, seem to express the highest levels of VDR and FXR. And so this raised the question of whether secondary bile could have an immune modulatory role by signaling through these secondary bile acid receptors. And to address this point, we analyzed the expression of genes involved in inflammasome. So the question is, how does this happen? How does antibiotics treatment result in enhanced inflammation and diminished antibody responses? Conceptually, you can imagine two distinct models. One is that you could argue that there's systemic access of PAMPs, bacterial products, caused by increased permeability of the gut epithelium. The second, you could argue there's an indirect effect of the microbiome on the immune cells in the periphery. So, to address the first question of systemic access, we measured the concentration of flagellin and endotoxin in the plasma and observed, in fact, there was no difference in controls versus antibiotics-treated people. This argues against a role for systemic access of these PAMPs. To address the second question of whether there might be an indirect effect, we performed an integrative modeling of multi-omics data sets, much like what I had mentioned to you before. And so we were able to integrate transcriptional profiling data with the metabolomics data, with plasma cytokines data, and the cell population. The results of our integrative analysis is shown in this slide. And in this network, what you can see is that the antibody titers shown on the top right and the change in inflammation shown on the left seem very distinct. They have very different connectivities. You see, the antibody titers are connected with metabolic clusters shown on the bottom right, largely, and some bacteria, whereas the inflammation modules are connected with some metabolic clusters, but also with secondary bile. In other words, there's a very tight inverse correlation between lithocholic acid, which is one of the secondary biles, but I could show you any number of secondary biles, and inflammation. But really, there's very little or no association between secondary bile and the antibody titers. Rather, what you see that is associated with antibody titers are metabolic clusters involved, enriched in fatty acid metabolism. This suggests that fatty acid metabolites might be controlling the antibody response to vaccination. And fatty acid metabolites might be the intermediary between the gut microbiota and antibody responses to vaccination. This is a hypothesis that we are thinking about testing in mouse models. So, to summarize, I think what we've learned from this study is that, in fact, the gut microbiota does have a profound effect on the immune system in humans, that as far as the adaptive immune response is concerned, in subjects with low baseline titers, that is, relatively weak immunological memory, the microbiota does act as an endogenous adjuvant. In other words, it promotes vaccine responses, antibody responses. In addition, we see that the microbiota, through the production of secondary bile acids, can control excessive inflammation. And so antibiotics treatment results in depletion of the gut microbiota, results in diminution of secondary bile, which ultimately results in an increase in inflammatory response. So this is what I've just summarized. The question we now have to ask is, what is the impact of the microbiome on immune responses to primary vaccination in adults or neonates and infants? As you can see, vaccines represent incredibly powerful probes on the human immune system. You can perturb the human immune system with vaccination, apply the tools of systems biology to analyze the signatures induced by vaccination, perform bioinformatics analysis, and generate interesting new hypotheses about the mechanisms by which these immune responses are generated and maintained. We can test these hypotheses using animal models, and eventually the understanding and insights that we gain from these mechanistic studies should and could inform future vaccine design. The particular example I've shown you in this presentation is our work that began by analyzing the human immune response to the seasonal influenza vaccine, in which we discovered that the expression of toll-like receptor 5 was induced and highly correlated with the subsequent hemagglutinin titers. We then tested the causal effect of toll-like receptor 5 and the antibody response to flu vaccine in the animal model in mice, and we discovered that in fact TLR5 did play a critical role in the immune response to influenza vaccine, and in fact this was because of its sensing of the gut microbiota. We then performed a subsequent study in humans to analyze the impact of the gut microbiota on immune responses to vaccination, and in that study we discovered that bile acid metabolism, in other words secondary bile acids, were critically involved in regulating the innate response. This has become a project in the lab in which we're investigating the impact of secondary bile on immune responses to vaccination, and perhaps in the not too distant future, in the next couple of years or so, we can think of us and perform an experiment in humans to evaluate the impact of this bile acid pathway on immune response to vaccination. So let me conclude by thanking the people who did the work in my lab. This was led by Thomas Hagen and Mario Cortez, two outstanding postdoctoral fellows. As I mentioned, this was a close collaboration with Nadine Rupail's lab at Emory, as well as with Galit Alter's lab, Hannah Golding's lab, Patrick Wilson's lab, and Andy Gowitz's lab, with generous funding from the Human Immune Profiling Consortium at HIPSI. Thank you for your attention.